Welcome to This Explainer. Today, we're unpacking the absolutely fascinating science of genetic engineering. We hear these terms thrown around all the time, on the news, walking down the grocery store aisles, in medicine, but today, we are going to actually look under the hood. We're going to discover how manipulating the literal, invisible building blocks of life creates some massive real-world impacts. So, let's get into it.
To kick things off, I really want you to think about this question for a second. How do we turn microscopic bacteria into life-saving medicine factories? I mean, it sounds totally like science fiction, right? Programming a tiny microscopic organism to just manufacture complex human medicine. But here's the crazy part. It's happening right now, all over the world, literally every single day.
Okay, let's establish our baseline definition so we're all on the same page. We are talking about literally moving the biological blueprints of life from one living thing to another. So genetic modification is the transfer of a gene or a specific section of DNA from one organism into the DNA of another organism. Think of it quite simply as a biological cut-and-paste job.
Here is our roadmap for this explainer. 1. Nature's scissors and glue. 2. Vectors, delivering the code. 3. Manufacturing human insulin. 4. Engineering our food. And 5. What does transgenic mean?
All right, Section 1, Nature's scissors and glue.
You can honestly think of genetic modification as kind of like a kindergarten arts and crafts project. First off, we need our scissors. In this case, those are restriction enzymes. They're used to snip out the exact required gene from the original DNA. And since different restriction enzymes target different DNA sequences, scientists can be incredibly laser precise. Next up, well, we need our glue. This is an enzyme called DNA ligase. Its entire job is to just stick these newly isolated fragments of DNA together into a single unbroken molecule.
Now, when those biological scissors cut the double-stranded DNA, they actually leave behind these short sections of single-stranded overhangs. We call these sticky ends. And they're called sticky because, well, if you take another piece of DNA that was cut with that exact same restriction enzyme, their bases are going to be perfectly complementary. They are basically primed and totally ready to lock together like puzzle pieces.
Moving on to Section 2, Vectors, delivering the code.
Okay, so we have our cut and pasted DNA. But how do we actually deliver this new code? Well, we use naturally occurring delivery systems to get our newly tailored DNA exactly where it needs to go. We primarily use two types of vectors for this. Plasmids, which are these tiny little circles of DNA found inside bacterial cells, are brilliant for transferring DNA into bacteria or yeast. Viruses, on the other hand, are perfectly adapted to transfer DNA into human cells or bacteria. And once it's inserted, this new code spreads like wildfire as the host cells multiply. So when our isolated gene and an open plasmid vector finally meet, it's a perfect match because both were cut with the exact same restriction enzyme. Remember those sticky ends? They fit together flawlessly. Then our DNA ligase steps in, acting as the glue, fusing them to form a single, unbroken recombinant plasmid. And just like that, the whole shebang is now fully prepared to be inserted right into a new bacterial cell. Let's hit Section 3, Manufacturing Human Insulin.
Before this technology existed, diabetics actually had to rely on insulin extracted from the pancreases of pigs and cows. It was a super slow, really expensive process. But step by step, genetic engineering completely changed the game. First, we isolate the gene for insulin production directly from a human chromosome. Second, we cut that human gene and a bacterial plasmid with the same enzyme to ensure we get those matching sticky ends. Third, we glue them together using DNA ligase. Fourth, we insert that newly recombined plasmid into a bacterium. And finally, we place those bacteria in a fermenter, where they multiply and just pump out pristine human insulin.
I really want you to think about the sheer scale of this journey for a second. We start with a tiny, truly microscopic plasmid, and we scale that all the way up to massive industrial vats of purified insulin. Every single new bacterium created in that fermenter expresses the human insulin gene. It's basically serving as its own microscopic pharmaceutical factory. Millions of lives have been saved, and it's literally all thanks to microscopic scissors and glue. Now, you might be wondering why on earth do we use bacteria as the host? Well, fundamentally, they actually read the exact same genetic code that we do, so when we insert a human gene, they read it perfectly and produce the exact same protein a human body would produce. Plus, doing it this way completely bypasses the ethical concerns of using animals, because, well, bacteria don't feel pain or distress. And structurally speaking, because plasmids are completely separate from the main bacterial chromosome, they are just incredibly easy for scientists to isolate and manipulate.
Moving right along to Section 4, engineering our food. By inserting foreign DNA into the genomes of plants, we've developed some pretty incredible innovations. Scientists have actually engineered crops, like maize, to contain a bacterial gene that naturally produces a poison. This poison is fatal to insect pests, which drastically improves crop yields. They've also created herbicide-resistant crops. This is a huge deal because it allows farmers to spray for weeds without accidentally killing the food itself. We even have things like golden rice, which is specifically engineered to produce the precursor to vitamin A to help fight nutritional deficiencies worldwide. And of course, there's drought tolerance, which allows crops to survive and thrive in incredibly harsh climates.
Now, looking at this objectively, these innovations bring some very distinct advantages. Reduced chemical pesticide use and increased yields definitely make farming more efficient and, in many ways, much more environmentally friendly. But we also have to look at the other side of the coin and weigh the very real disadvantages here. The high costs of these specialized seeds can really shut out poor farmers, and it can create a cycle of dependency on the specific chemicals manufactured by those exact same seed companies. Furthermore, there are ongoing ecological debates. There are unknown long-term effects on local insect populations and wildflowers that scientists are still actively studying today.
And our final topic, Section 5. What does transgenic mean?
You've probably heard these terms interchanged, but there's a key difference. A genetically modified organism, or GMO, is simply an organism that has had its genome altered. But here is the kicker. This can absolutely be done using DNA from the exact same species. However, the term transgenic refers specifically to an organism that's been modified with DNA from a completely different species. So if you modify a plant using DNA from its very own species just to enhance a certain trait, yep, it is a GMO, but it is absolutely not transgenic.
I'll leave you with this final thought to ponder. As our mastery of genetic engineering scales up from microscopic bacteria creating insulin to entire global food networks designed to withstand climate change, how will we choose to balance its incredible potential against the mysteries of rewriting life itself? The tools are literally in our hands, but the future of this science depends entirely on how responsibly we decide to wield them. Thanks so much for joining me for this explainer, and keep exploring the incredible building blocks of our world.